So, welcome, my dear friends. So, today's our eminent resource person, Dr.O.Padmaja Madam GARU, Assistant Professor, Department of Botany, Tara, Government Degree and PG College, Autonomous, Reddy. Madam, please welcome. Madam has huge experience regarding of plant sciences and plant biotechnology. So, Madam, please welcome and share your screen and start your session.
Thank you, Tirupati sir, for giving this opportunity. Screen sharing, sir. Screen sharing.
ఇచ్చాను ఒక్కసారి మళ్ళీ ఒకసారి చూడండి. Yes, madam. Clearly.
Sir, okay, sir.
All right. Clear, madam, please proceed.
Okay, myself, Dr.Padmaja, Assistant Professor of Botany, Tara Degree College, Reddy. We are going to start today the continuation part of this embryology. Yesterday, we thought that yesterday we completed the class of flower, a reproductive part, and the essential and non-essential organs, and regarding the microsporangium, male gametophyte, and development of male gametophyte, and at last the end products of the two male nucleus are formed in the pollen tube of the male gametophyte. And today we are going to start the megasporogenesis, megasporangium, megasporogenesis, and female gametophyte. So we are going to cover the contents like megasporangium, megasporogenesis, development of female gametophyte, matured embryo sac or matured female gametophyte, fertilization, and conclusion. Yesterday, same like that only, we completed the microsporangium, microsporogenesis, development of male gametophyte, and formation of the pollen tube and the male gametes. So today we are going to start the megasporangium. Before entering into that, we have to know the idea regarding the female reproductive organ that is known as the gynecium. Yesterday we thought that male reproductive organ as the andrecium. So this is the diagram. The side, the diagram is showing that this is an essential organ and the fourth whorl of a flower that is known as the gynecium. And the gynecium consisting of three parts. The basal part, we already studied that the cushion part of the flower is known as the thalamus. And thalamus is present at the base. On the thalamus, the ovary, style, and stigma. These three parts combines to form called as the gynecium. And gynecium is also called as female reproductive organ. And it is also consisting of a tip known as the stigma. And between the stigma and the ovary style is there. And the basal solan part is known as the ovary. Inside the ovary, ovule is present. And now, the free unit of gynoecium is called the pistil. It is also called as the pistil, and it is also called as the carpel. And what is mean by carpel? We already studied in the gymnosperms, in the lower plants, instead of reproductive organs, they are called as microsporophils and megasporophils. Microsporophils are known as androecium, megasporophils are known as the gynoecium. That means the flower is also formed from the vegetative part. The previous embryology is saying that the flower, the reproductive part, is the modification of the vegetative leaves. And so it is considered as a carpel. The carpel is also known as the megasporophil. The sporophil means the fertile part which is having the sporangia. They are called as the sporophil. If they contains the megasporangium, it is called as the megasporophil. So this carpel is also known as the megasporophil. And the carpel is differentiated into three regions: style, stigma, and ovary. The end of the carpel receives pollen grains is called as the stigma. And the stigma will be of different shapes in different families or in the different plants. It may be of hairy-like structures in a Fabaceae, or it may be a capitate-like structure, or it may be a sticky-like structure. There are different types of formation of stigma will be seen. And somewhat the style will be very much elongated, or somewhat it will be a less long, narrow tubular structure present between the stigma and the ovary is called as the style. So the basal zonal part of the carpel is called as the ovary. So now here the carpel, megasporophil, and the gynoecium is the same terminology using for the same structure. And next one is the ovule. So the ovule is also called as megasporangium, where this ovule is present. The ovule is present in the ovary. The ovary is the ovary is the basal part of the gynoecium, and this ovary, inside the ovary, ovule is present. The other term we are used, the ovule is as a megasporangium. And when we see the structure of the megasporangium, what are the parts present in the megasporangium? The stalk of the ovule is known as the funicle, and each ovule is attached to the placenta by a small stalk. That means every structure will having the stalk. The stalk of this ovule is known as the funicle. And the funicle, the place of attachment of funicle with the main body of the ovule is called as the hilum. And each ovule is consisting of integuments. There may be a one integument or the two covering the ovule. And they are called integument and the inner integument.
Madam, excuse me, your voice is not clearly audible, madam.
And sorry, ovule is also called as megasporangium. Now, if you see the structure of megasporangium, the stalk of the ovule is known as the funicle, and each ovule is surrounded by the integuments, outer integuments and inner integuments. If only one integument is present, it is known as the unitegmic ovule, and if it is consisting of two integument, it is called as bi-integument ovule. Now, in some Gamma-Petalae and Archiclamidiae, the nucellus is teninucellate. That means, what is meant by nucellus? Nucellus, it is a fertile tissue that is present in the integuments, or the nucellus is embedded by the integuments. The nucellus is or of two types. The nucellus will be of two types, teninucellate and crasinucellate. In such ovule, the innermost layer of the integuments become the specialized to form the nutritive function. So, in some of times, regarding the nucellus, we are going to ask the question also. And the nucellus, the body of ovule consisting of central solid parenchymatous mass of cells enclosed by the integuments. That means this nucellus is a tissue that is enclosed by the integuments, and from this nucellus only giving rise to the megasporangium mother cell. That means the megasporangium mother cells are forming from the nucellus. The one of the cell will be functions as megaspore mother cell. And now the ovule. So the ovule, the main concept is ovule is known as the megasporangium. Each megasporangium consisting of funicle integuments. Inside the integuments, nucellus is present, and nucellus is giving rise to the megasporangium. And this nucellus is of two types. One is crasinucellate and teninucellate. Crasinucellate means if the nucellus is present in much amount, if there is a massive, the more layers of the nucellus is present, that type of nucellus is known as the crasinucellate. It is mainly present in the polypetalae and the monocots. And such ovules are called as the crasinucellate ovules. If the nucellus is more amount or more layers of the nucellus is present, that type of nucellate, prosellate nucellate it is called. It is mainly present in the polypetalae and monocots. Teninucellate means only a single layer of nucellus is present. That is called as a teninucellate. It is mainly seen in the Gamma-Petalae. That means when we compare the the polypetalae is the advanced one, when primitive one when compared to the Gamma-Petalae. And inside that nucellus, one of the cell will functions as megaspore mother cells. These megaspore mother cell undergo reduction division. As a result, 4 megaspores are formed. Now the question is, the nucellus is haploid in condition and the embryo sac is, nucellus is diploid in condition, embryo sac is haploid in condition. The nucellus undergo reduction division. As a result, it gives rise to the megaspores. From the 4 megaspores, the 3 will be disintegrate, only 1 will be functional. That functional one develops into the embryo sac. So the other name for embryo sac is the female gametophyte. That means the formation of these megaspores from megaspore mother cell is known as megasporogenesis. And each embryo sac consisting of the nucleus we will see here. That means each ovule, that means the embryo sac is present in the ovule. And these are all the parts present in the ovule. So hypostage. Hypostage is a cup-like tissue that is present below the embryo sac. Below, if it is present in below, it is called as hypostage. That tissue is these are the structures. These are the special structures seen in the ovules. And according to Whitnam, the hypostage limit the expansion of the embryo sac into the chalaza. So the growth of the embryo sac will not be extended. So it is acting as a limiting factor here. And Johnson said that is responsible for maintaining water balance in the dormant seeds also. That means the seeds, in order to undergo the germination, the water is very essential. These are also helpful for the storage of this water. And Baumann produce hormones required for the embryo sac. These are some structures seen in the ovule. And epistage. It is the cup-like cutinous tissue formed above the embryo sac. The structures present above the embryo sac. If the structure present below the embryo sac is called as the hypostage, and it is nutritive in function. And another one is the actuator. Any ovule structure associated with the directed growth of the pollen tube towards the micropyle is generally referred to as obturator. This obturator is helpful for the pollen tube to entering into the embryo sac. That means it is showing just like a direction to the entry of the pollen tube. Yesterday, we also thought that the entry of the pollen tubes are of three types: orogamy, chalazogamy, and mesogamy. Whatever may be the entry, but the pollen tube will be entering into the embryo sac through the micropyle and then releasing its male gametes. Now, here the types of ovules. These types of ovules, regarding this type of ovules, it will be related to the taxonomy also. And here 6 types of ovules are seen here. That the 6 types of ovules, in that first one is the orthotropous ovule. It is a primitive ovule, and it is also called as the straight ovule. In gymnosperms, we are seeing these types of ovules. Mainly in cycles, we are seeing the orthotropous ovule. And here the ovule, what are the parts of the ovule means? One is the funicle and the hilum, chalaza, and the micropyle. These are all remaining in a straight line. That type of ovule is called as the orthotropous ovule. And another one is it is present in the polygonum. Is a polygonum. In that polygonum, the first studied embryo sac is, or the typical embryo sac is studied in the polygonum. And the anatropous ovule, it is totally inverted ovule, and it is also called as the advanced ovule. That means in family Asteraceae, we are seeing this type of ovule, anatropous ovule, and the body of the ovule becomes completely inverted, and the micropyle lies towards the funicle. So it is mainly seen in the gamopetale members. And next, hemitropous. In hemitropous, the body of the ovule is placed transversely at right angles to the funicle. The micropyle and the chalaza lie on one straight line, is seen in the ranunculaceae. And campylotropous ovule, the body of the ovule is curved so that the micropyle and chalaza do not lie in the same straight line. It is seen in the leguminaceae family. In leguminaceae, the ovules are all of campylotropous ovule. And amphitropous ovule, the curvature of the ovule is very much pronounced, and the embryo sac also become curved. It is seen in the Butamaceae. And amphitropous ovule, the curvature of the ovule is very much pronounced, and the embryo sac becomes curved. Here, just like the horse-shaped structure of the embryo sac is seen in the amphitropous ovule. And Allismaceae and Butamaceae. And Surendered ovule, this type of ovule, here the funicle is forming like a spring of a watch. And here seen in the girophytic members like Opuntia. And these are the 6 types of ovule. And if you see these are the diagrammatic representation of the ovules. And in this, mainly the types like anatropous means straight ovule, total inverted is orthotropous is straight ovule, total inverted is anatropous ovule, and campylotropous ovule is present in the family Fabaceae. And the Surendered type of ovule is seen in the Opuntia. And the straight ovule or the primitive ovule, these are seen in gymnosperms also. And now, megasporogenesis. What is meant by megasporogenesis? Formation of megaspores from megaspore mother cells is known as the megasporogenesis. And where these megaspores are present. The nucellus, the tissue that is present inside the ovule, nucellus give rise to megaspore mother cells. The nucellus is a diploid, megaspore mother cell is a diploid in condition. This megaspore mother cell undergo meiotic division. As a result, 4 haploid megaspores are formed. This formation is known as megasporogenesis. This total process is taking place in the ovule. And the megaspores are arranged in the linear form. In these, the 3 megaspores all will be degenerated, only one will be functional. The development of female gametophyte or the embryo sac. The embryo sac is haploid in condition, gametophyte is haploid in condition, and the megaspore is the first cell of the female gametophyte. Yesterday class, the microspore is the first cell of male gametophyte, or the pollen grain is the first cell of male gametophyte. Here the megaspore is the first cell of female gametophyte. During megasporogenesis, 4 megaspores are formed in the form of a linear tetrad. Of these 4 megaspores, the upper 3 degenerated and disappeared. The chelazal megaspores become functional and develops into an embryo sac. This type of structure will be seen in the polygonum. And the nucellus of the functional megaspores divides by the mitotic way and each megaspore consisting of haploid nucleus. And these haploid nucleus, as a result, by one mitotic division, 2 nucleus are formed, and these 2 nucleus will be moved to the polar micropyle and the chelazal. And these, again, each nucleus at the polar divide twice, forming the 4 nucleus. As a result, 4 nucleus are present at the micropylar end and 4 nucleus are present at the chelazal end. And now the embryo sac. The embryo sac consisting of, out of 4 nucleus found at the micropylar end and the embryo sac, 3 organized into the egg apparatus. That means in an embryo sac, there are 8 nucleus are present. In that 8 nucleus, 3 will be arranged at the micropylar end, and one of the nuclei of the micropylar end will be moving to the center of the sac. And from the chelazal end, 3 will be remain. They are acting as the antipodals, and from that, one be moving to the center. They are called as polar nucleus. These polar nucleus combines to form a fuse to form a diploid secondary nucleus. These diploid secondary nucleus giving rise after fertilization to the endosperm. So the embryo sac described below is developed from 1 megaspore. Hence, it is described as a monosporic embryo sac. In a polygonal type of embryo sac, they are consisting of egg apparatus, the nucleus, and the antipodals. The 8 nucleated embryo sac is commonly known as the polygonal type of embryo sac. It is the primitive type of embryo sac. It is the first discovered embryo sac. This is the whatever we thought just now, total matter is in the form of diagrammatic representation. First you see, this is the megaspore. This megaspore mother cell is formed from the nucellus. This megaspore mother division, as a result, 4 megaspores are formed. That division is known as the meiosis. We know by the meiosis 4 haploid nucleus are formed. In these 4 haploid nucleus, upper 3 will be disintegrating or they are disappearing. These are black color structure or disappearing. Only one will be functional one. This functional megaspore enlarges in size. The nucleus is haploid. These haploid nucleus undergo first mitotic division. As a result, nucleus are formed. These two nucleated haploid nucleate megaspore undergo another division. As a result, four nuclei are formed. And these four nucleate megaspore undergo another mitotic division. As a result, total 8 nucleus are formed. In this total 8 nucleus, 3 will be remained at the micropylar end and 3 will be remained at the chelazal end, and in between the 2 will be function and fuses to form a secondary polar nucleus, and later they give rise to a matured embryo sac. So the final diagram is the matured embryo sac. Where this matured embryo sac is present, it is present in the megasporangium, that is ovule. If we see the structure, so the opening end of the ovule is known as the micropylar end. In this micropylar end, the integuments leaving the space, this is called micropylar end, and opposite to micro, and inside the integuments nucellus is present. These nucellus give birth to megaspore mother cells. These megaspore mother cells undergo reduction division. As a result, 4 megaspores are formed. In these 3 disintegrate, only 1 megaspore is functional. In that 1 megaspore having a haploid nucleus, this haploid nuclei undergo 3 mitotic division. As a result, 8 nucleated embryo sac is formed in polygonum. And this is the structure of a matured female gametophyte or the embryo sac. In angiosperm, the female gamete is called as the embryo sac. It is 7-celled and 8 nucleated structure. Related to this question, this matter, they will asking the questions like, well, which type of embryo sac is seen in the polygonum? It is a 7-celled and 8 nucleated. That means 2 nucleate fuse to form the diploid polar nuclei. So it is considered as a 7-celled, 8 nucleated structure. It consisting of 3 parts. They are the upper micropylar end consisting of 3 as the egg apparatus, and 2 are called as the antipodals, and the center it is called as the central cell. Egg apparatus, it is present towards the micropylar end of the embryo sac, and it consisting of 3 cells. 1 is egg and 2 are synergids. On either side of the egg, synergids are present. These synergids having a finger-like filiform apparatus which are helpful for directing the pollen tube to enter the embryo sac and releasing the male gametes into the fusion. And egg, this is called as the female gamete. It is the central cell of the egg apparatus. It is completely surrounded by its own wall. And on either side of the egg, there are synergids. There are elongated cells present on either side of the egg. Each shell show a nucleus at the upper end, the vacuole at the lower end. So this is the reverse to the egg. If you see the egg, it show a large nucleus towards the lower side and the vacuole towards the upper side. But in synergids, vacuole at the lower side. And the apical region of each shell shows longitudinally oriented fiber-like structures. These structures are called as the filiform apparatus. They are finger-like projections seen in the synergids. And it may help in the absorption and transport of food materials from the nucellus to embryo sac, and some special chemicals, and helps in the entry of pollen tube into the embryo sac. And then the antipodals. The antipodal cells are present toward the chelazal end of the embryo sac. They are 3 in number. They are smaller than the egg apparatus, and they are short-lived and degenerate or epipodals. That means they may be degenerate of after some time. They are not participating key role in the fertilization. And central cell. It is the largest of the embryo sac. It shows a large central vacuole and two haploid nucleus. Two haploid nucleus fuse together to form a diploid secondary nucleus. The fusion takes place at the time of entry into the embryo sac. And now this is the structure of matured embryo sac, whatever we saw just now. So this is the micropylar end. Micropylar end is having the egg apparatus. Egg apparatus consisting of two synergids and one egg. And if you see the synergids, they are having the filiform apparatus that are helpful for the entry of tube. In the center, the two polar nucleus combines to form the diploid secondary nucleus. And it is also called as the central cell. And at the chelazal end, three antipodals are present. And whenever the enter into this, is that one of the main gamete will fuse with the egg. Another main gamete will fuse with the secondary polar nucleus. As a result, the fusion between the main gamete and egg ures to zygote. The fusion between the another main gamete and the secondary polar nucleus ures to the endosperm in the angiosperm. And after fertilization, will be disappearing and the will be disappearing. And fertilization. Sexual action is called as the fertilization. During pollination, the liberated pollen grains are carried to stigma of the same flower or different flower by different agents. The pollen grains will contain the vegetative cell and the generative cell. The generative cell again divides to male gametes and germination of the pollen grain. The pollen grain absorbs the secretion of the stigma and increases in size, and the pollen tube will be enlarged, and this pollen tube will entering from total size and reaching the embryo sac and releasing these male gametes into the embryo sac. And later, the intent grows out through the germ pore in the form of a long tube called the pollen tube. Normally, one pollen tube arises from a pollen grain, but in some members like Althea and Malva, many pollen tubes are formed. That condition is called as polycyphonous condition. And, we also studied that the entry may be of different types. One time they asked the question like, which family the chelazogamy type of entry of pollen tube will be taking place. So just remembering the chelazogamy C and the cucurbitaceae, we can easily remember for the like writing the objective type of bits. And whatever may be the entry of pollen tube, there may be a porogamy, chelazogamy, and the mesogamy. Porogamy through the micropyle, the pollen tube entering into the micropyle, chelazogamy by the entering into the chelaza, and the mesogamy by the integuments. Whatever may be the entry, but is entering into the embryo sac to the micropyle only. Irrespective of the reason where the pollen tube enters the ovule, the pollen tube grows through the nucellus and finally enters the embryo sac at the micropylar end. And now here the pollen tube having the male gametes and how the fertilization is taking place. The pollen tube has a terminal and the pore is formed that the tube nucleus is helpful for the directing the male gametes into the pollen tube and entering into the ovule. Through this pore, the pollen tube discharges two male gametes and some amount of cytoplasm. So the fusion of gametes. Here, one of the male gamete will fuse with the egg, another male gamete will fuse with the secondary polar nucleus. So there are two times the fusion will be taking place and two times the fertilization will be taking place. So in angiosperm is considered as a double fertilization. There are One is first fertilization is known as the syngamy. As a result of syngamy, one male gamete fuses with the egg to form a diploid zygote. So that is called as the zygotic fertilization. And another one is known as the triple fusion. Triple fusion means already the secondary polar nucleus consisting of two nucleus and another male gamete combining and fuses with the secondary polar nucleus. As a result, three nucleus are participating in the fertilization. So it is also called as the vegetative fertilization, and here the three nucleus are participating, and it is called as the triple fusion. So syngamy means one of the male gamete, one of the two male gamete fuses with the egg to form the dip zygote. This is called a syngamy, and it was first reported by Strasburger. You see here syngamy S, Strasburger S. So he is the person who reported in the year 1884. It may be considered as a bit in the entrance. So the male gamete fuses with the egg. As a result, a diploid structure zygote is formed by the fusion known as the syngamy. It was first studied by the Strasburger in 1884. And next one is the triple fusion. Triple fusion means second male gamete in the pollen tube fuses with the diploid secondary nucleus that is present in the embryo sac to form a triploid primary endosperm. That means one of the male gamete fusion leads to the zygote, another male gamete fuse to leads to the formation of endosperm. It is also called as the primary endosperm. In abbreviation, it is called as the PEN. The chromosome number of PEN is 3N in condition in angiosperms. So three nucleus are participating in this, so it is called as the triple fusion. It was reported by Nagashin in 1898. And if you see the equation, male gamete fuses with the two polar nucleus. As a result, a three nucleated endosperm is formed. But what is the function of this endosperm? It is a nutritive in function. It is providing the nourishment for the developing zygote to form into embryo. In angiosperm, two types of fertilizations are recorded in the same embryo sac. In one embryo sac, two times fertilization is taking place. So it is called as double fertilization. If you see the diagram here, it is called as the double fertilization. Here, the two times fertilization is taking place. So total structure is taking place in the ovary. Whenever in the gynecium is taking place, and later on the stigma and the style will be disappeared, and this total structure will be developing into the fruit and the seeds after fertilization. And this total process is taking place in the ovary of the gynecium. And here we are clearly seeing the embryo sac and the ovule. Inside the embryo sac, egg, antipodals, synergies, secondary nucleus, and the entry of the pollen tube, the releasing of the sperm cell. Here the pollen tube consisting of two nucleus. One of the nuclei will fuses with the egg. Another male gamete will fuses with the secondary polar nucleus. As a result, here you clearly understand in the third diagram. Here there is a fusion is taking place. As a result, zygote is formed here, and here the endosperm is formed here. Here the two times fertilization is taking place in a single embryo sac. It is called as double fertilization in the angiosperms we are seeing. As a result of this double fertilization, zygote is formed and the endosperm is formed. This zygote will be developing into the embryo, and this endosperm giving the nourishment for the developing embryo. So at last, we can come to know that megaspore undergoes sequential meiotic and mitotic divisions. Only one of the megaspores develop into the embryo sac within the ovule. in most of the plants, while other three will be disintegrating. So today we completed, if we see the brief regarding our topic, first gynoecium, the gynoecium consisting of ovary, style and stigma. Inside the ovary, ovule is present. The ovule, if you see the structure of a ovule consisting of a stalk known as the funicle, integuments. Inside the integuments, nucellus is present. These nucellus give rise to megaspore mother cell. These megaspore mother cell undergo reduction division. As a result, megaspores are formed. In these 4 megaspores, 3 will be disintegrating. Only 1 will be remaining. In the remaining 1 megaspore, only haploid nucleus will be there. It is undergoing 3 mitotic division. As a result, 8 nucleated structure embryosac is formed. This is the overall synopsis of today's class. Thank you.
Madam, thank you very much.
Thank you, sir.
Okay, madam, thank you.